afternoon. It's now my pleasure to introduce our next fireside chat guest, Faraz Ali, CEO of Tenaya Therapeutics. So again, the format for this session is roughly a 25-minute fireside chat. And if anyone in the audience has questions during the session, feel free to type them into the question box and we'll try to ask them on your behalf. So Faraz, thanks again for joining us. And to start off, maybe you could give us an overview of Tenaya's manufacturing and CMC capabilities and how that fits into Tenaya's broader strategy.
Yeah, thanks, Agola. Thanks, as always, to Chardan for the invitation here. I do want to first start with an acknowledgement of our Chief Technology Officer, Taehyung Kim, who I'm standing in for him today. He's an incredibly important member of our team, taking a well-deserved vacation. First executive I hired when I came here as CEO in 2018, and recently promoted to Chief Technology Officer, taking a well-deserved vacation. So I think to get into Tenaya's manufacturing strategy, I think it's important to first remind people what is your manufacturing strategy has to follow your corporate strategy. It doesn't stand in a vacuum. So just initially articulate or re-articulate for those who are familiar with us, our corporate strategy, and then how does manufacturing fit with that? So importantly, Tenaya is a company that's focused on the heart. We're addressing both rare and prevalent forms of heart disease with gene therapy as well as regenerative medicine approaches. We are technically modality agnostic, so we look at AAV gene therapy as well as other modalities, including small molecules that obviously influences our strategy. Part of our strategy is to internalize some core capabilities, including novel capsids and capsid engineering. We have an intention to become a fully integrated company that can launch and commercialize some of our own wholly owned gene therapy products. And we do intend to be a partner of choice, both for large biopharmaceutical companies who are interested in gene therapy for the heart, as well as from early stage academics who may be sitting on some promising ideas. So that's us in a nutshell. We're about 120 people in South San Francisco, and we went public last year. So how does that influence our strategy? Given that strategy, we thought it was important early on for us to internalize both non-GMP and GMP capabilities for AAV-based therapies. So we made the decision very early on to internalize non-GMP capabilities, including upstream and downstream process development, analytical development, quality control, from everything from shake flasks up to the 200-liter scale. We also made the decision about 2 years ago to also internalize GMP capabilities. And so we're going to, I know we'll talk more about that later, but we are going to internalize that as of this year. We're on track to have a fully operational facility this year. For non-AAV-based therapies, though, that's where I think there's more nuance. I think for small molecules, which we have in our earlier pipeline, those are very well characterized as very strong capabilities in the CDMO universe that we can rely on for production of small molecules at the GMP scale. So we don't intend to internalize that. We intend to outsource that. And for other modalities, I think it's really going to depend. The devil's in the details and the nature of the molecule, whether it makes sense to internalize that and use our current non-GMP and GMP capabilities for those, or to outsource those as well. It really depends on the nature of the modality. So that's us in a nutshell.
Great. And so can you expand on what you think sets your manufacturing approach apart from others in the cell and gene therapy space?
Yeah, well, look, I mean, in some ways, we're not alone. We're not alone in internalizing non-GMP and GMP capabilities. Perhaps we've chosen to do both earlier in our product life cycle and early in our corporate history than some other companies. But you know, that's one thing that's different. And I would say we're probably among a handful of peers who have chosen not to plant their GMP flag in some completely different state far across, the country. We decided to put our GMP facility right here in the San Francisco Bay Area, reasonably close to our research labs and our PD labs, and that was a deliberate choice. I would say what maybe makes us a little bit more unique is the huge emphasis we have put on and a focus on scalability of our manufacturing processes very early on in our development, partly because of our intent to be able to address larger indications. We need to be able to produce product at large scales to be able to address the demand for larger clinical studies, and we need to be able to get to lower cost of goods. So that emphasis early on in our thought process to think with the end goal in mind, I think sets us apart a little bit, and we've already made some choices, strategic choices as a result of that, including investing in research for novel capsids, and then therefore also looking at manufacturing platforms get allow for that scalability as we need to go to bioreactor scales that are beyond 500 liter, beyond 1,000, beyond 2,000, maybe even 5,000 and 10,000. We're not making those investments in those bioreactors today, but we are making the investments in the processes today to be able to address that novelty and to be able to achieve that scalability. So I think that's perhaps one of the things that makes us somewhat unique compared to our some of our peers at this stage in the development.
Great. And so you mentioned, internalizing GMP capabilities. I understand you have your GMP facility that's expected to come online this year. So can you discuss a little bit around the timelines there and why you felt it was important to have it come online in this timing right now, and also about the capacity that you touched on a little bit, but would love to hear more about that.
Yeah, sure. So first off, I'll just reiterate the guidance that we give to all of our investors, recognizing there may be some investors in your audience here. We made a public commitment that we expect to have a fully operational cGMP facility by the first half of 2Q22. We tend to keep the promises we make and make promises we can keep, and so we're on track for that guidance. We also expect to file INDs for our 2 lead programs by the second half of this year. That includes TN-201, which is a gene therapy product, and TN-301, which is for a small molecule product. Again, the TN-201 will be produced at our cGMP facility. TN-301, small molecule, will be outsourced. So we are on track with all of the guidance that we've given to investors. And so the first half of this year, we will have a fully operational cGMP facility right here in the San Francisco Bay Area. In terms of capacity, so we are building our manufacturing facility in a 94,000 square foot space. We acquired that space back in 2021. Initially, we're only building out 50% of that space, and we're keeping that second half in strategic reserve. And our initial manufacturing design is it's a very modular design based on pods. So what does that mean? While our initial cGMP capacity will be modest, bioreactors at the 200 liter and 1,000 liter scale will be up and ready to go on day one. But it gives us the opportunity to expand our capacity rationally as our pipeline matures. So we will have in this space, and given the design we have, both to scale up as well as to scale out rationally as new products come into the pipeline and come into focus. So that's part of what went into our thinking. In terms of commercial, this initial footprint with modest modifications, we think will be commercial ready if and when we get to that moment in response perhaps to an accelerated development timeline if we have some really compelling data. So we're already thinking with that commercial end in mind and believe that this current facility and in this initial footprint can be made commercial ready with modest modifications. So why did we do all that? I think it just these choices that we've made to internalize non-GMP and GMP capabilities, gives us several advantages. Obviously, and I'm sure you've heard this from others, more control over cost, more control over our timelines, more control over our quality of our products. I think we have more flexibility to respond to external and internal events, whether there's been an evolution in the FDA requirements for manufacturing, even early stage manufacturing. So we are better positioned as a result of controlling our own destiny with non-GMP and GMP capabilities to adjust to those FDA requirements. We're better positioned, I think, now to respond to external perturbations like, say, COVID, as well as to respond to both opportunities for acceleration or setbacks in our own portfolio versus having to negotiate with a CDMO every time there is either something that's slowing down or something that needs to accelerate. So we have that flexibility to respond to events. And finally, and I don't think this can be stated enough times, we have a deeper understanding of our own product, its product attributes and the product performance and the drivers of those attributes and performance. And so because we've internalized the processes, because we've developed our own processes, because we've developed the assays, not only the required assays, but also the ones that may be more unique to our products, we just have a deep understanding of our products. And I think that's that's a pretty important thing when you're dealing with gene therapy in potentially, hopefully, transformative therapies that are going to touch people's lives. So those are some of the advantages of the approach that we've taken. I'll pause there.
Yeah. Great. So kind of picking up on what you were just mentioning in terms of the of understanding your product, I think you mentioned earlier that Tenaya is laser focused on heart disease. So can you expand on what this means in terms of learnings across your different programs and implementing learnings regarding manufacturing as you advance your pipeline?
Yeah, I mean, one thing that comes with heart disease is just the fact that you have to be prepared for larger indications. I mean, heart disease is the leading cause of death in the world, more than all oncology combined. That one benefit we have is that we're able to understand the genetics of it and to go after genetically defined subpopulations within this very large space. But even the rare diseases within heart disease tend to be large. Our leading program, TN-201, is going after the leading genetic cause of hypertrophic cardiomyopathy, and our estimated prevalence is more than 100,000 patients in the U.S. alone. And then for TN-401, where we're going after the PKP2 mutations, that's a leading genetic cause of arrhythmogenic right ventricular cardiomyopathy. And again, our estimates there are more than 70,000 patients in the U.S. alone. So even in the rare universe, these are large indications compared to some of the ultra orphans that some of our peers are pursuing. So just the fact that we're going after large indications means that we need to be able to scale up and to be able to prepare to deliver drug for larger clinical studies potentially. It's also, I think we have to be, because heart disease has been a space that has been dominated by small molecules for large indications. The mindset is different when it comes to the expectations of costs and safety and therapeutic index. And so I think that also is something that we have to take into consideration. So that's partly why scalability with the lens to also reduce cost of goods to be able to supply drug reliably for large indications. I think all of that flows with the focus on the heart, that we need to be prepared to go big, to be able to drive down costs, have pristine, product attributes, no impurities. We can't afford to get those things wrong. So that really puts a higher pressure on us to get manufacturing right early on in our life cycle and early on in our corporate history.
Got it. So to that point, are there particular innovations that you're looking at that can address or circumvent some of those key cost drivers associated with manufacturing for gene therapies?
Everything, so you'll hear me say it, it'll become boring after a while, right? It's scalability so that you can actually produce the drug reliably in larger quantities necessary to go after larger indications, but also to be able to drive down costs and to be able to do that, to be able to have more control over your cost of goods, to be able to have more flexibility in where your pricing is eventually for a rare disease versus for a more prevalent indication. I also think just overall, we need to find ways to reduce dose, and so we need higher potency per infused capsid. We need the industry needs that as a whole. We certainly feel that we need that for what we're doing. We've now understood that there is such a thing as dose-limiting toxicities associated with AAV. And so, above 1E14 vector genomes per kilogram, people start to get nervous. When I say people, that includes my peers in industry as well as the regulators. For a 100-kilogram patient, that's a 1E16 vector genome dose. That's like 10 quadrillion viral genomes, right? So we need to find a way to make sure that we're finding ways to reduce dose. And that means fewer empty capsids, for example. That means each capsid has a maximal chance of being packaged well, unpackaged well, transduce the cell, unlock its payload, express that protein reliably, and with very, very low likelihood of inviting an immune response. Manufacturing has a critical role to play in that equation, every step of that equation. Being able to produce the amount with fewer capsids, with high potency and low immunogenicity. And so it's really an important part of the equation. I think your process is your product. And so manufacturing really needs to be looked at as, an important aspect of your product attributes. So in terms of innovation, anything that drives ability to scale up, anything that improves your ability to have fewer empty capsids, anything that improves your ability to have lower immunogenicity. Those are all important attributes. And there's no single silver bullet innovation, honestly, Gula. I think there's a lot of different things that we need to continue to optimize on as an industry and as Tenaya for its own products.
Got it. So as you mentioned, some of these issues regarding AAV safety have been of great interest to a number of stakeholders in the space, including regulators. So what do you think is of prime importance in terms of working with regulators, particularly regarding CMC issues? And is there lessons that you can take from other gene therapy programs that have run into CMC challenges?
Yeah, look, the FDA has already said it. I'm sure some of your other guests have, mentioned the famous quote of, in the case of gene therapy, they think, 80 to 90% of what they focus on is manufacturing and 10 to 20% of the product. Maybe that's an overstatement, but it's important. The FDA clearly understands the importance of manufacturing, and rightly so. I think what we've seen is, for every clinical hold that appears to be related to maybe the product and product's attributes, some of those clinical holds, there's another clinical hold that is purely about manufacturing, and the FDA not having the confidence that the changes you made at this stage results in a product that's performing at the next stage. And so I think we have to, bottom line is, the big picture is you need to know your product, and you need to understand how changes in your process affects your product attributes and then in turn your product performance. You need to be able to convey to the FDA that you understand and understand those things, and therefore you have a plan that is well considered and well laid out. What does it mean, and what do we learn? We've seen so many promising therapies from our peers sometimes stumble. Nobody's had to stop a program, but programs have certainly been delayed at everything from preclinical to clinical, or phase 1 to phase 2, or phase 3, and then getting to commercial. So what are the things that we've learned? One is that's partly has influenced our choice to internalize our production processes, and so that we really understand what we're doing, including, very importantly, and often, as I said, underappreciated, assays to allow you to really understand your product and your product performance. So we've chosen to internalize those early on in our history. I'd say the other thing that we've tried to do, or we tried to do at least, and have successfully done so far with our early work, is to lock in your process early, right? So that you're minimizing really the risk of changes from the tox lot that was used for your GLP IND enabling studies to what you're producing GMP for first in human studies to what you're producing later for later studies and eventual commercialization. If you can come up with a process that you think you can lock early, then you minimize those changes across different transitions. And I think that's good for you, that's good for the regulators, ultimately it's good for your program. So that's something that we try to do. We may not always be able to do it on every single program, but certainly on our first program, that's what we've tried to do. Lock the process early, and then use that same process that we use in IND enabling studies for first in human studies. And I think the other thing that we've chosen to do that is a little bit different, I mentioned early on, we decided to plan our GMP facility in the San Francisco Bay Area, instead of putting it out in Texas or North Carolina, which is a popular destination. And part of the insight there is that, our process development labs are right next to our research labs. So there's a very strong crosswalk and crosstalk between our research organization and our manufacturing organization from the get-go. They're producing the vector that's going into the research studies. They're the ones scaling up to 200 liter for the tox studies. But then those same people who developed the process that we locked for our GLP studies are the same people who can just get in a car and drive across the bay and enter our GMP facility and make sure that we're scaling up to the 1,000 liter cGMP run. Having the same people on both sides of it, and having these facilities only 30 minutes away from each other, I think in the short term and long term will prove to be a strategic advantage in terms of ensuring that continuity of know-how from all the way from your shake flask and early production all the way to cGMP. So that was a strategic choice we made, partly by seeing what we've seen in the industry and setbacks in the industry.
Got it. And so just taking a step back, and maybe this is something that you just touched on already in your answer to the past question, but what do you see as the biggest CMC challenge in gene therapy right now, and any solutions on the horizon?
So, a fair question, and I think I'm going to try to respond in two different ways. One is it's a technical challenge, right? Manufacturing this live medicine at the scales that we need and the quantity we need. This field is still in its infancy. We're still a very long way from being able to just say, hey, I'm just going to toss over this product to a CDMO and they're going to produce it, in a very routine way. We are still in the infancy of this field. And so, I think there's been a lot of emphasis put on the capacity challenges, but I think we can solve capacity challenges, right? CDMOs are out there, they're buying up capacity, they're consolidating. And so I think the capacity problem is easy to solve. The technical problems are not as easy to solve, right? And we haven't seen the same kind of, I'd say, innovation ecosystem emerge to say we're going to achieve breakthroughs on the manufacturing side to allow for scalability, to allow for this innovation to become more portable and modular, so that it becomes routine. So we need more innovation on that side as well, on the technical side, and we need a deeper bench of talent to do that, both on the capacity, production, but also on the technical innovation. So I'd say that's one of the big challenges. But the other challenge that doesn't get a lot of airtime, honestly, is the business model, right? What we're dealing with is disruptive innovation, right? We're going from a world of potentially chronic therapies for large indications to potentially bespoke, one-time, quote unquote, curative therapies for smaller genetically defined populations that eventually we hope to also apply to larger indications. But the mindset of a different product profile, I don't think has yet come all the way into saying, like, how does the manufacturing facility, supply chain, how does all of that need to be reconfigured to adjust to a world where your product is a potential cure? And so instead of building up to these large, farms of bioreactors like Genentech has, north of here, that may make sense for large chronic indications for, large indications that are chronically administered therapies, but it may not make sense for, a rare disease. And so if you're going to have 5 different rare disease products in your early history, there's going to be these peaks and troughs. So how do we design the facility? How do we think of our talent strategy? How do we think of our, supply chain? How do we think of our entire operating model to adjust this novel modality and this novel way of treating patients? I don't think as a field, we've put that much thought into it, into what does that need to look like in steady state in the future. So I think that's a challenge. Until we think through all of that, we may still be making suboptimal choices today.
Right. And so to wrap up in the last few minutes of our session, I wanted to get your perspective on what do you think investors might be missing about Tenaya's manufacturing capabilities today?
Well, I mean, I could say with confidence that our early investors didn't miss anything. In fact, that's, the Series A and B investors supported the strategic decision we made 2 years ago to invest in a CGMP facility, and the Series C investors, who ended up being crossover investors to a T, that was part of their diligence. They were asking, like, what are you going to do about manufacturing? And all of them were on board when we said we intend to internalize we have internalized non-GMP, we intend to internalize GMP. So they formally, when they wrote those checks, they knew that what they were signing up for, and they knew that some of their money was going to go not towards the product innovation side of things, but also go towards the manufacturing side of things. And they fully supported that. As a public company now, we have a broader base of investors, including people who may not know us as well. They may not fully appreciate the strategic importance of manufacturing, that essentially you have to think of manufacturing as part of the product attribute, that this is a major investment to eventually have a product that has certain performance characteristics, and the importance for us to continue to invest and innovate in this space versus rely on somebody else. But that burden is on us to educate the newer investors about why we made the choices we've made, why we continue to invest in this. So I would say overall, though, we have a very, very supportive investor group. They may not appreciate the depth to which we're doing things, and that we feel forced to do things because of the unique corporate strategy that we're pursuing. But I'd say that's an opportunity, not a problem.
Great. So on that note, we're at the 25-minute mark, so I'd like to thank you again, Faraz, for your participation, and we'll talk to Faraz again in a couple of hours, in a couple of hours at our panel that will discuss large manufacturing considerations for larger indications. So more to come on that.
Always enjoy the opportunity. Thank you.